Get started. Good morning, everyone, and good day. And welcome to this webinar on validation and non-composted soil amendments and crop inputs. This is being jointly hosted by Arizona LGMA and California LGMA. I'm Greg Comer, the Technical Director of the California LGMA, and I'm joined by Vicki Scott, who is my counterpart at the Arizona LGMA. We are also joined by an esteemed panel of subject matter experts who will be presenting the different topics on the agenda today. Before introducing the panelists, I want to let everyone know that we will be recording this presentation for future use. I want folks to know that as we go along with this presentation, please add any questions or thoughts you might have into the Q&A box in your screen. So we will be having a nice Q&A discussion at the end of the panelists' presentations, and we've allotted a good amount of time for Q&A during this particular webinar. So please, as the presentations are going on, you have some questions, please add those into the Q&A box, and we'll get to your questions during that moderated session. So let me move forward a little bit here. This presentation is going to be going for about an hour and a half or so. So we've scheduled it from 9 to 11:30. We can go a little bit over, but some of our panelists do have a hard stop at 11:30. So if there's still some Q&A going on after that time, we do have some extra time that we can allot to that. But just know that we are planning this for about an hour and a half. And again, we do want to answer any questions and all questions if possible. So we will be able to go a little bit over if that's possible. So the agenda for today is really to talk about validation as it pertains to non-composted soil amendments and crop inputs. And so I'll go into a little bit here in a second why non-composted, but the real idea is we want to go over our just basic LGMA requirements for soil amendments and crop inputs that are not compost-based. We want to talk about what is validation. So the big take home for all of us today is we do have requirements in the LGMA for demonstrating that products being used on your farms come from a validated process. But we also understand that a lot of folks don't necessarily know what that means or what to even present to an auditor when that question comes about. So that is the real big take home for this webinar. Is we brought together these panelists, these folks who really understand this process very well, just to allow you to get some basic understanding of validation and the difference between validation and verification. But also, really, we want you to know after this presentation is over, or this webinar is over, what are some of the take-home things that you should be thinking about when you get your documentation from your supplier? So that's really the big take-home message for today's webinar. We want to talk about process authorities and give some validation examples and have a nice, robust discussion about the validation process and how we got to where we are today. We're going to have the supplier perspective on validation. That's obviously a really key part of this. Not only do the farmers need to understand what it means to have validated products and to apply validated products, but what does that mean from a supplier perspective, and how, as a user of these products, can you work with your suppliers to make sure you're getting what you need from them? As I mentioned, we want to have a nice panel discussion with some robust Q&A if possible. And then Vicki and I will wrap it up with some take-home messages and take-home thoughts for, again, what you should be thinking about when you're getting some documentation, or when you're actually buying products, what you should be thinking about or considering for a validated product. For our presenters today, as I mentioned, esteemed presenters and folks that have just a plethora of understanding and knowledge about this subject. To start off our presentation today, we're going to have Channa Rock talking about, give us an intro to validation. Channa Rock is a professor at the University of Arizona in the Department of Environmental Science, and also maintains a joint appointment as a water quality specialist with UA Cooperative Extension. Dr. Rock is a primary investigator on several projects related to microbial evaluation of water quality for the protection of public health. Dr. Rock currently supports the California-Arizona LGMAs by providing science-based recommendations for ag water treatment under the new irrigation water metrics, as well as supports the Arizona Department of Agriculture in disseminating the FDA's Food Safety Modernization Act Produce Safety Rule growers trainings. Chana holds a BS degree in microbiology from the New Mexico State University and an MS and PhD in environmental engineering from Arizona State. And I will just add to her bio that her understandings regarding the principles of validation have been a very critical aspect for our industry-wide implementation and rollout of the LGMA's irrigation water treatment requirements. So, while Chana doesn't necessarily deal with the soil amendment side, her perspective on validation, what it means, how to do it, is going to be a really critical part of this presentation, and we hope that you will find it extremely valuable. Mr. Cliff Coles is a recognized process authority for the Almond Board of California and has food microbiological experiences that cover a span of almost 50 years. Sorry, Cliff, kind of aged you there.
Yeah.
Until his recent partial retirement, Mr. Coles founded and was the president of Consulting Microbiology of California Incorporated, CMC Inc., an organization that I've used quite extensively over my many years in the industry, where he has been actively engaged with the fresh produce industry in multiple capacities, including being an expert witness, performing validations in conjunction with the Preventive Controls Rule of FSMA, reviewing food safety programs for the industry, providing expert counsel during recalls and outbreak investigations, providing training and outreach, and providing input on multiple industry microbiological related task forces and committees. Mr. Coles and the laboratory CMC Inc. remain as the microbiological microanalytical authority for the Del Monte Foods Corporation. Mike Menace, for the past 12 years, has been the VP of Food Safety and Technology at True Organic Products and also supports the regulatory and R&D segments of True. He has been involved in various levels for organizations including the Organic Trade Association, United Fresh Produce Association, California Organic Products Advisory Committee, and most recently the Center for Produce Safety and Arizona LGMA Food Safety Committee. As a former director of a food safety laboratory, Mike has a unique perspective regarding testing methods, laboratory capabilities, and food manufacturing facilities. The principles in this knowledge base have been applied to the needs of organic fertilizers under the construct of LGMA and FSMA. Mike received his PhD in chemistry from the University of Louisville. Mike, his wife, Janelle, 3 kids, 2 dogs, and a bunny, have called California home for at least 20 years. Opportunity and the beauty of this great state have brought them here, and the produce from California Ag has kept them here. And then wrapping it up, no introductions are really needed for this guy, but Dr. Trevor Suslow is an emeritus professor and extension research specialist whose career spanned 30 years at UC Davis and in industry. Dr. Suslow helped found and worked at Advanced Genetic Sciences and DNA Plant Technology Corporations from 1981 to 95 before joining UC Davis. At UC Davis, Dr. Suslow had a research and outreach education program that had statewide responsibilities in quality and safety of perishable horticulture commodities that spanned pre-harvest to post-harvest. His research combined lab and on-farm packing facility research on pathogens of concern, including E. coli, Salmonella, and Listeria in conventional and organic production systems, for the purpose of identifying opportunities for optimal microbial reductions and delivery of safe products to consumers. Currently, Dr. Suslow runs a food safety and quality consulting business where he's still heavily engaged with the fresh produce industry. Dr. Suslow received his BS in agriculture with honors and a PhD in plant pathology from the University of California, Berkeley. So I do appreciate all of you for being here today, and we definitely understand that, Vicki and I always talk for Vicki, when we have questions or have problems, who do we call? We call these folks. So definitely appreciate that you're here with us today. So as I mentioned at the beginning, why are we here in the first place? So the LGMA does have requirements regarding validation. Under the new requirements that were passed in August of this year, we do have some new areas and new categories for soil amendments and crop inputs. However, the area of processed soil amendments and crop inputs, what we call category 7C, have always actually been part of the LGMA metrics since its inception in 2007. But one of the changes that we made this year is that we've just expanded what we mean by processed products. And so with that expansion came questions regarding what does it actually mean that these products are actually validated? And when I say we're not including composted products, and that the reason for that is because composting in general, from a FSMA perspective, from the LGMA's perspective, the process for creating compost and the documentation and the requirements that we've had for compost are I shouldn't say necessarily well understood, but they're definitely better understood than the requirements for validation for processed products. And so that's why we're not really discussing composting today. we really wanted to focus our attention on those processed products. So think about it, something made in a facility that doesn't have a natural process for its creation, like compost. We're talking about things that go through some sort of a treatment process, like a heat step or a chemical treatment, etc. Examples may include chicken pellets, blood meal. You can read the list here. There's quite a few products that this could potentially fall under, but at the end of the day, what we're really concerned with is for processed products, do they have a validated process for reducing pathogens of concern? And that's why we're here today, to make sure you guys understand what this potentially looks like and what it means for you. Let's see here. So under the metrics, as I mentioned, we've had requirements for validation for quite some time. And as it pertains to this, to validation, under our metrics in what's called our table 3 for soil amendments and crop inputs, validation is the treatment processes applied to the soil amendments and crop inputs shall be done via a process validated to assure the process is capable of reducing pathogens of human health significance to acceptable levels. And so again, what we're looking for when we want to get your documentation for validated products is that the products have gone through a process capable of reducing pathogens of human health significance to a quote unquote acceptable level. So we'll go through some of that language here today. My computer has frozen, so give me a second here. There we go. Now, as far as, what is the specific requirement when we get audited, this is the requirement that we have for our handler members and for growers that are supporting our members. All test results, certificates of analysis, and documentation shall be current, reviewed before use, and available for verification. So when you think about, from a handler member perspective, our industry, when you get an LGMA audit, what is the expectation that you're going to deliver to the auditor when they ask, do you have documentation that supports the product you're using is validated? This is where that requirement comes from, right? It's a very simple, basic requirement. But over the years, we've had this requirement since 2007. But as we're starting to review some of the documentation that's actually available to the industry, we found it really important that we have this discussion today, because we're noting that some of the information being provided to our industry just doesn't meet, what we call the general requirements for a validated process. And so one of the things I want, before we get into Chana's intro to validation, one of the things I wanted to really quickly go over is this language that we have in our metrics that says, we have this area in table 3 called the rationale for processed products. And we've had this in the metrics since 2007. And the rationale for the requirement for validation stems from this language. The US FDA has established the validity of D values and Z values for key pathogens of concern in foods. This method of process validation is currently acceptable to US regulators. Alternatively, results of an inoculated test pack utilizing the specific process is also an acceptable validation of the lethality of the process. So that has been in our metrics since 2007, but I would, Vicki and I, we've discussed this quite a bit, and even recently with our auditor training, this came up. What does this exactly mean? And what is the expectation from the industry? So real fast, what I want to go over is just what are D and Z values so that the industry has just a general background about it. And then from there, like I said, our panelists will go into a little bit more detail about what does all this lingo mean, and how is it how practically can it be used by you folks? So a D value is the time required to destroy 90% of the vegetative cells or spores at a given temperature, which is also called the decimal reduction time, usually referred to as the D value, okay? The D value usually varies inversely with temperature. So if you think about the ability to destroy 90% of vegetative cells, as temperature goes up, the time to do so usually goes down, and vice versa. As the temperature goes down, the time to kill 90% of vegetative cells usually takes longer. The Z value is equal to the increase in the number of degrees from a given starting temperature that results in a 90% reduction in the D value. So, you're going from a reduction in vegetative cells under the D value to a reduction in the D value with the Z value. So it's like, whoa, this is really confusing. What does this all mean? Simply, the Z value is the number of degrees of temperature change necessary to change the D value time by a factor of 10. And I have just a little demonstration. Again, I'm not expecting everyone to quite get all this just from these couple of slides, but this will be available for you to study later if you want. But from the figure that's available, this is an FDA type figure. As you can see on the figure on the right, and the amount of time it takes at 222 degrees, it takes 10 minutes to reduce a certain amount of population by 1 log, all right? So D222, that's the 222 degree D value. It takes 10 minutes at 220 degrees Fahrenheit to reduce a population of microbes by 1 log or 90%. Now, when you increase the temperature to 240 degrees, remember that inverse relationship, right? At D240, it takes 1 minute at 240 degrees Fahrenheit to reduce the same population of microbes by 1 log. So at 220 degrees, it takes 10 minutes to reduce the population by 1 log. At 240 degrees, it takes 1 minute. So you can see a 10-fold reduction, right? The Z value then is just a simple mathematical equation that you subtract 222 from 240. So if it takes 1 minute at 240 and it takes 10 minutes at 222, you just subtract 222 from 240, and the Z value for this particular reduction is an 18 degrees Fahrenheit Z value. So that's all that means. Again, it's a lot more complicated when we start talking about it, and we'll get into that a little bit with Trevor and Cliff and with Chana's introduction, but I just wanted to quickly review some of that stuff with you folks to have a background for the next presentations coming up. So I'm going to stop sharing this, and Chana, you're on. Thank you so much.
All right, thanks, Greg, so much. So I appreciate you guys all joining today for the opportunity to discuss a bit more detail about validation and some examples. So as Greg said, I come from the water world, and so a lot of my examples will be really based in how we're looking at some of these agriculture water treatments, but the concepts transfer kind of across the board. And so I'm going to review the differences between validation and verification. I think a lot of times we get folks that interchange those two words, and they're very different, and they're both very important. And then that will kind of lead into the rest of the speakers today, giving some real world examples and kind of walking through some of the expectations for some of the products that you all are using. So again, kind of overall, here's what we want you all to learn from the day. Obviously, understanding the difference between validation and verification. We're going to talk about some of the activities that are tied to these different areas. And I'm also going to include some monitoring efforts, because monitoring fits under this umbrella of verification. And a lot of you are very familiar with monitoring efforts in different aspects that you conduct currently. And then I'll just walk you really quickly through an example from agriculture water treatment perspective. Okay, so validation and verification. And so I'll just kind of run through some of these. You guys will all have access to these slides, so you'll be able to review in a future date. So validation really asks whether the hazard analysis, so that specific hazard we're talking about right now, right, are those pathogens of human health concern, was complete, and if the control measures were effective. So, are we designing a process to reduce, as Greg just talked about, the D and Z values, to reduce those human health pathogens to an acceptable level for that product to be used in your farming operation. Alternatively, verification asks whether the system is being implemented according to the plan. So were there any deviations from the process that was laid out for validation? And if there were, you would pause and stop and say, well, this isn't a validated process. And this isn't, we're not doing what we're saying we're doing to produce the product that's most appropriate for that reduction in human health pathogens. Another way to look at it, validation with a goal, is to demonstrate that that process, again, when operating within those established limits, produces a product consistent of specified quality with a high degree of assurance. So when we're looking at validation processes or treatments, for example, we want to have this high degree of assurance and this trust that if done correctly, you're going to get the same result over and over and over again. And really, the validation of a treatment process is necessary to, again, attain those soil amendments or those products, or for example, if we're talking about water treatment, with that desired quality attributes. So again, in this case, it's making sure that we're seeing that reduction in human health pathogens. Verification, alternatively, is again that process of confirming the truth. And oftentimes We also think about, and we all learned, way back in grade school, maybe, the idea of the difference between accuracy and precision, right? So hopefully you guys have all seen like the target, drawn up on the board. And the difference is between, accuracy, like hitting the target, and then precision, hitting the repeated or close to repeated same spot over time. And so if we think about a valid treatment, that would be one that's scientifically accurate or correct, right? The process, we've proven it, and we've proven that's been able to control those hazards that we're concerned with. The verification, alternatively, is to make sure that it's implemented precisely, or that the steps in the system are repeatable over time, so that you have trust in that process, you have trust in that treatment. And so really, to achieve that optimal treatment, or to achieve that product that you're looking for, you have to both have a valid and a verified process for that. So what do validation studies involve? And we're involved at some level in looking at validation of agriculture water treatment technologies or chemistries. But again, they're similar for whatever products that you might be thinking about. So they're scientific studies. They're usually industry adopted standards or others, technical review. Oftentimes they're performed by the manufacturer or the supplier, and should be for the most part. Oftentimes you can also find folks like myself conducting benchtop laboratory validation studies, or third-party laboratories being able to do that. It's also important that if you're looking at the results of a validation, or the write-up, the SOP for a validation, that those studies should really target those foodborne pathogens of interest, specifically for your commodity or those indicators. So again, things that we think about for bacteria, Shiga toxigenic E. coli or Salmonella. For protozoa, it might be Cyclospora, although that's super challenging for reduction in Cyclospora. You might also see things like indicator organisms. We talked about that a lot in the water world, and specifically looking at generic E. coli or total coliforms, and linking those to on-farm uses and or treatments. So again, just to kind of reiterate, those target organisms for validation should either be those human health pathogens, indicator organisms, say like generic E. coli or total coliform, for example. But we also have a group of organisms identified as index organisms, and these might be modified pathogens or pathogens themselves that have been tested and validated in a variety of different settings, and ones that we can kind of go back to the literature to understand their relation to human health pathogens, and have been successfully used for validation studies. One thing to note, because we're concerned with human health pathogens, any validation using plant pathogens we would consider not sufficient. And really that's because we don't know that plant pathogens behave or die off the same as human health pathogens. And that could potentially create some questions or some issues about, again, that validation process. I think it's also important for you all to think about, validation is just beyond that bacterial inactivation. So obviously we focus really heavily on those human health pathogens, but there's other considerations that go into a validation, and that's really understanding those soil amendments or crop inputs, or that water treatment chemistry, and the conditions that surround that validation. So some of the examples of things that might also be included in a validation are assessment of different times. Greg just talked about time and temperature. Those are super important. Potentially pH, if it depends on the process, for example, used to reduce those microorganisms. Potentially the percent moisture within that product, and also considering, well, what are we starting with and what is the aim that we're ending with? So thinking about biomass or organic matter content, different things like this. And, it's really, sorry, I gotta move something around. It's really, important to think about, what is the process that you are going to be used, or what is the environment where these products are also going to be used for you all, and how that may or may not impact what we might see or hear from that validation. So switching a bit onto verification, I touched on this before, but verification is really those activities other than monitoring that can establish the validity of the process and that you're following the SOP and that you're operating according to that plan that you've established for the reduction of those organisms. So some of the elements of a verification, and again, this is things that you're going to want to potentially look for or ask about a validated process. So are they calibrating the monitoring devices? So it's great that you are, collecting temperature data, but it's no great if that thermometer is not working appropriately. So, thinking about the timing in which some of these monitoring devices are calibrated and making sure that they are being calibrated and that you have documentation of that. So really reviewing those calibration records. Again, that record review of any type of monitoring records that might be collected during that validation process, as well as if there were any corrective actions as a result of a deviation from that process. Additionally, thinking about verification, when and how they're performed. And again, this is usually on equipment or instruments used for monitoring or verification. It's really up to the supplier to determine at what frequency to ensure the accuracy of those measurements and being able to ask questions and being able to get information back from a supplier about, what frequency they're using. And really by checking accuracy against those standards at the condition that the instrument will be used. So again, making sure that you've seen folks set up a validated process in which they're defining their limits. What is the upper temperature limit? What is the lower temperature limit? And making sure that those equipment that are being used are calibrated within those ranges of those, specific temperatures, for example. Another part of verification, again, and this is probably the most important part, is this record review component. And so again, thinking about monitoring the activities that have been done for a validation, how frequently are they performed? Is it every 30 minutes? Is it every hour? Is it only one time? And making sure that they have a definition for that and a justification for that. Again, when any corrective actions have been performed during monitoring, that might indicate a non-compliance for those critical limits that have been set. And then again, that they've been reviewed, dated, signed by this, whomever is in charge, supervisor, responsible party, and that you all have access to that, or at least, if you had questions about it, they'd be able to produce that for you. So most of you, if you're on the growing side, are super familiar with monitoring aspects, right? And so monitoring is just defined as conducting a planned sequence of observations or measurements to assess whether that process or procedure is under control and to produce an accurate record. And really monitoring is a part of that verification, right? So you're putting all this data into that. So without adequate monitoring, there's no way to know, or at least in this case, to prove if you're meeting those critical limits and operating in an appropriate manner. So again, if the control measures are being taken to manage the risk, or that hazard, for example, if they're being met, and really kind of thinking about what that might involve for monitoring. So what are they monitoring? How often, how are they doing that? What types of tools are they using? What's the frequency that is monitoring is being conducted, and ultimately who is in charge of that? So they're all important components of monitoring. I think oftentimes we think about the what we're monitoring, but we don't spend enough time thinking about how and frequency and who. So some of the how, oftentimes, at least from a water perspective, we're thinking about physical or chemical measurements. And we like to lump these into two different areas, and we want these all to be quantitative. I mean, we're getting, values back related to whatever monitoring we're doing. And so we group them into what we call operational limits and then critical limits. So operational limits are going to be a way, potentially a high and a low value of whatever parameter you're monitoring. So say example, residual parts per million, ppm of a chemistry. And that's going to trigger a process adjustment if you go above or below those operational limits. So for example, that might be you increasing the dose of a chemistry. Alternatively, your critical limits are outside of that window of your known validation. So say you know that you can't drop below a certain residual and still achieve that log reduction of bacteria that you're looking for, that's going to trigger a corrective action. And really those critical limits are super important from a validation standpoint. We recommend that observational data, because it's not quantitative, is not super recommended. It can be part of a monitoring process, but shouldn't be the only thing that you're relying on. And for some of these validated processes, you might see that some of the monitoring might be in real time. So they might have real-time sensors, pH probes, temperature sensors, things like that, that are tracking data over time. And I think that's important to think about as well. So examples of how and what to monitor, these are pretty straightforward. A timer, you're going to be looking at minutes or hours at a certain temperature. Thermometer, again, are you meeting those temperature requirements for your critical limits? pH, are you within your range? Your scales, and important really think about scales if they're calibrated appropriately for wet or dry weights. And then obviously the thing we're most familiar with is looking at the microorganisms themselves. So either a culture looking at the actively replicating organisms, or is it a molecular method? And I think it's also important to think about in a validated process, how they're also looking at those microorganisms and the methodologies related to that. So in summary, before we get into an example, validation and verification are super critical and essential to understanding the potential hazards and risks posed by the products you use. And then that records review component is super important. So, hopefully this gives you a couple of more things, a little bit more ammunition to be able to go ask the suppliers about the products that they're providing to you, and a bit more about their validation. So a current example of an EPA pesticide product to treat microbes for ag water. We all know that this is kind of pending, and that not all labels are currently targeted for human health pathogens. So we do have some independent studies that have been performed. We've done some validation work. Trevor has done some validation work, and others specifically looking at these products and how they are efficacious against foodborne pathogens and human health pathogens. So as a result of a lot of questions within industry, FDA and EPA announced a validation protocol. And so here's the link. If you Google FDA, EPA, ag water validation, this will come up. Here's the link also, so you guys can take a look at that. But basically, they've outlined a validation protocol that looks at E. coli, Salmonella, and Listeria, multiple different strains of these organisms. It establishes time requirements, contact time, and it establishes temperature requirements. And it also establishes kind of initial water quality requirements or kind of ranges for that water quality. So keep an eye out for some of those changes to the labels, but this is a great example about how there was not a validated process specifically for these products, and now there is one. So we'll see what happens with the changing of some of these labels. So how folks are doing validations, and this is just an example from ag water. Say they're looking at free chlorine concentrations in a treatment system. They're collecting water samples pre and post treatment, and they're trying to document the reduction of coliforms in E. coli. So they're trying to determine a % reduction. So you're looking at the number of E. coli in the untreated water versus the number of E. coli in the treated water, and then really using those values to calculate a % reduction. And so here's an example. We're starting out with 324 colony forming units per 100 mLs. In treated water, you're reducing that from 324 to 10.5. And you run through the calculation and you're looking at a 96.6 0.75 percent reduction, so a bit over 1 log reduction. It is important when we're thinking about ag water and the organisms that are used for a validation, for example, make sure that you have enough in your source versus after your treatment to be able to conduct a validation. So if you're not starting with very high population, you won't have adequate data to be able to conduct a validation. So once you know how your validation performs, kind of your lower and upper limits or your chemical limits, then you can use that treatment and be able to show that you can keep your values within that range on your farm. So again, if you're doing an on-farm validation, that's going to involve collecting data at a frequency that you decide to demonstrate control over those critical parameters. And here's just an example. Hopefully you guys have seen this before. We've presented before. We've got free chlorine on the left-hand side. We have time along the bottom. Our operational limit is 5, and then our critical limit is 2. So that teal line is residual of free chlorine. As you see, as it dips below the operational limit, that suggests a process adjustment. And then as we dip below that critical limit, we're outside of that window of that validation, and that's when a corrective action would be required. So once you kind of do that and understand how it works on farm, you can transition to kind of a normal operating procedure with SOPs that you develop. And really, we started to see this shift in industry better understand, how to do a validation on farm for some of these chemistries. And it's been exciting to see some of the data coming out of that. So again, last slide, here's some real world kind of example. A grower identifies a pathogen of concern, and they're selecting, a validated water treatment chemistry as an intervention. So they're applying the treatment at the same concentration, so residual ppm and temperature as what was conducted in the validation. They're documenting the application of the system, and they're measuring residual ppms of that chemistry. And they're assessing data following implementation. So they're looking at bacteria, pre and post treatment. And really what we would look at this is that this system is successful at controlling that hazard, both doing a validation on farm and then verifying all that information is collected appropriately. So I'll go ahead and end there. I know Trevor and the rest of the folks are going to be walking through some more real world examples. But we appreciate your time, and hopefully you guys have some good questions in a bit.
Okay, thank you, Shauna. With your permission, Greg, although I'm not going to wait for it, Shauna, actually, I think a question here for you would be appropriate while it's fresh in everyone's mind. And this is where we often get, slight differences of opinion about what's validation, what's verification. It gets a little messy. But in your example for on-farm validation of water tests, others might put that more in the verification category because you're sort of verifying the conditions that were validated for pathogens against indicators. Is that, the definitions really aren't that critical in terms of what you do, but I'm just curious if you had anything that you share about where you would see it fitting in.
Yeah, and I think we go back and forth, right? I think it's a little bit of a gray area. I think, obviously we want to be looking at the chemistries against the primary targets of human health pathogens, but we know we can't do that in an on-farm setting. And so we've kind of resulted in falling back on indicator microorganisms for the most part to be able to do an on-farm validation under real world conditions, but trying to go as close as we can to whatever we saw in the initial kind of benchtop validation as far as parameters.
That was the point, and that then ties into what I'm going to share. So thanks for answering it right up front while it's fresh in everyone's mind. So
Thanks, Trevor.
Let me give this a shot and see if that works. Hopefully you can see my screen now. And what I decided to do to share with the group today, and I really appreciate the opportunity to do that. And if you think back to the list that Greg showed,
Trevor, I'm not seeing your screen. Is anyone else seeing it?
Oh, sorry. How about
There you
now?
go.
Yeah, it would help if I would have hit share button. Okay. Is that there's just a long list of soil amendments, fertility management, stress, management, formulations, various crop inputs. So I decided within the time available just to pull out a couple of examples that span a long period of time, not as long as Cliff, but a better part of 40 years in the private sector, back at the university, and still ongoing. And I kind of put it in what I call sort of the watch list for growers and handlers as our primary target audience here. Things that you should be watching, and it's a bit different than what Shauna just presented, and that sort of verification, potentially on-farm validation process, is something that is in your hands, is in your court, and that you may commission that to be done, together with the monitoring and everything else. In this category, I don't see it as likely or as commonly unless something goes wrong, and then often growers, handlers, whoever may be doing some things in more of an investigative mode. So what I'm going to do is talk through a couple of different examples, and then leave some of the more thermally treated validation process controls to Cliff and Mike. There we go. So this is sort of my view of the sort of shared responsibilities to minimize produce safety risk, and it cuts across basically the handoff train. So I see there's a lot of the validation and the elements of process control go with the manufacturer. Depending on how things happen, there's I think the level at which the distributor has certain responsibilities, and then there's sort of the grower responsibility, or within that production supply side area. And breaking down, this is not an exhaustive list by any stretch, but I just wanted to try and point out again my view of what has always happened and what needs to continue to happen, and across the different possible soil amendment materials, it needs to be much more broadly applied. So that's really a validation studies that define the process, and that the process prevents pathogen survival of the raw material, the feedstock, whatever it may be, or contamination of the finished product that's then going to be sent out and marketed. And that includes handling and packaging that prevents post-process contamination. And again, we can if there's interest through questions, I think Mike probably will handle a lot of certainly what they do and how they look at it. But also, I think that there's a manufacturer responsibility for many of these products to conduct what I call end user risk validation studies, and I'll get back into that a little bit later. For the distributor, and again, I'm basing this a lot on what I've experienced, what I've seen, what we've done when we go out to do an investigative analysis for something that caused a widespread contamination of leafy greens that resulted in multiple acres being destroyed. That ensures the integrity of the delivered lot. Again, prevention of contamination of either bulk containers or the transfer process handling, if that's part of their responsibility. And ensures that the end user, which is the grower, basically is aware of the potential for pathogen regrowth or in-use contamination growth. Again, that is something that falls then into the grower responsibility. I think it's the responsibility of the grower to be informed of risk validation challenge studies that should have been conducted further up the line. And as Sean has said, it could be a commissioned study, by the manufacturer, possibly in-house, but usually it's better for it to be a third-party process, or it could be in a qualified academic lab, public, research lab, whatever form that is. And then, really be informed of the recommended and critical in-use preventive controls if the risk validation challenge study demonstrates that if contaminated, that product can support the growth of pathogens of concern. And here, of course, we're talking basically about bacterial pathogens. And then certainly to become informed of any recommended verification SOPs based on those risk validation studies. So, what might you verify? What might you monitor if that's part of it? That's atypical, but at least understand if there's something that clearly can be done and should be done to prevent contamination of the product at the use level, not at the production or distribution level. So, here's an example which comes up many, many times. So the assertion is that fish emulsion, and of course, I've picked a couple of the more commonly used materials, cannot be a source of foodborne pathogens. So why is that? Again, what floats around is that, well, they're heat treated, typically at 180 degrees Fahrenheit for X amount of time, filtration, which, may or may not be part of pathogen reduction, but, it would pull things out. But the other key step besides heat and time, and, we're not going to get into D's and Z values. Wow, Greg, but all relevant, but that's kind of hard first thing in the morning, huh? Is acidification with a variety of acids. Phosphoric acid is the typical one, and, to reduce putrefaction and rancidity, that's dropped to usually somewhere around pH of 3.5. Now, what you have to understand is some products are deacidified either partially or to neutrality because they're being combined with other things. And these days, a lot of that is beneficial microbes, competitive microbes, or it's meant to stimulate their growth. And the claims are, or the evidence is, that within their growth, they're producing antibacterial metabolites of different groups and classes to actually prevent pathogen growth should a cross-contamination event happen, and therefore it's all protected. So if we look a little bit deeper, and this is pretty highly simplified process flow for a variety of products that are out there, and you can easily go in and, well, not easily, you can go in and look at some of the different formulations and what they're based on. And there are other publications that help walk through it. But just kind of going from left to right, there's the heat treated, acid stabilized, and then what I think, there's a value. The watch list question is, has the pH been raised for various reasons, yes or no? Then there's others that are simply acid stabilized, trying to achieve the same thing but without heat, and then they may or may not raise the pH again for the end user claims and purposes. There's other processes which are basically enzymatically digested or hydrolyzed and then heated and then acid stabilized. Those products tend to stay at that really low pH. And then there's another group which would be, a fermentation with a starter culture of lactic acid bacteria, and they generally have to add a nutrient, a carbon-based nutrient, and it's often molasses. It could be other things. I didn't want to put all the possibilities in here. And the pH is then raised, and then they're often adding these proprietary cultures and then adding additional molasses as a growth media. So what's important then is to look across the finished process pH that's reported in the technical sheet, and I'm not going to name specific products here, but you can see the range. And some of the issues go back to what is a validated process, what is the process control, what are you monitoring? My opinion would be, in say product spec B, that they can have between a pH of 3.2 to 4.5, which is a pretty broad range. We'll come back to this, but one would be pretty lethal to things like Salmonella, E. coli, and others. They won't grow, but they're also not going to die off completely should a contamination event happen or if it's incomplete. Others, as I said, they raise it to neutrality for the purposes of the benefits that are claimed to go along with the product, and others, they may only report a pH level at the use rate, which again brings it closer to the range which many of the human pathogens we're concerned about would have the potential to grow and compete. So I'd say one of the watch list questions, just something to have in mind and ask the supplier, try and get at it, because these things are typically not validation studies that a grower or handler is going to run, unless there's a problem. And then it's really more, was this product contaminated or not? But if it's done, usually they obtain a single routine lot per formulation. The more effective approaches would do multiple tests over a period of time to look at that accuracy and precision, that consistency to deliver. And I know Cliff will talk quite a bit more about this, so I'll move on. And then to inoculate the product with one or more challenge pathogens. And again, these days it's kind of grown from 1 to 2 to 3, including E. coli, even Listeria, typically at a fairly high level, something you wouldn't expect to see at the farm level, but it's because they want to have this quantitative, log reduction, and then either recovery after single or multiple time points. And all of these things make a difference depending on the formulation. So 30 minutes versus an hour versus 24 hours. And it revolves around, again, pH. Competitor growth products can be part of the validation study. You have to give it time for them to do their job, and then you report on the log reduction. So now this is a very busy slide. I'm going to walk you through it, but this is why I said, there are times when a quick and dirty sort of, on farm or by firm validation study, if there isn't information, can help you, because if you do a quick and simple one and there's no lethality, then that right off the bat. But it is complicated and it is complex, and this may sound fairly self-serving, but you have to really get into the weeds. You have to get into the details to know what you might expect as the end user. So in this case, picking a variety of strains, I just using Salmonella. Here in the undiluted fish emulsion, you can see, because of the pH of this particular formulation, you get a very significant, very quick lethality to isolates if you're growing them in a typical lab bench type of validation study. They're not stress adapted. You can demonstrate, oh look, it really works really well. They die off a lot. If you pre-adapt them, using a variety of stresses, and what happens with things like Salmonella, whether you pre-adapt them to low pH conditions, not the pH of the fish emulsion, but pretty close, or even desiccation or drying, things that might happen, you have still lethality, but it's far less under the conditions of the study than you would if you just threw some nice, happy cells that were just recovered from culture. If you look across, if you just use a single strain, let's just say you use Salmonella Newport, which is often, one of the outbreak strains, you would think that the sensitivity, even stress adapted, would be pretty high within that formulation. But as you move to some of the gold standard for doing these kinds of validation study, the Semptenberg, it still, has lethality within the time frame, but it's far less. So hopefully you're able to follow all of this. A different type of formulation that we've worked with in the past, these algal extracts plus some enzymes plus some microbes, undiluted. It's not growing, but it's really not particularly lethal to an introduced challenge isolate in a validation study. So again, you understand what that's about, and the same, with whether they're stress adapted or not. If they're not stress adapted, and at the tank mix for fish emulsion, you can see at the conducive temperature, you can get quite a bit of growth. So this is what you're looking for, and it's really not different in stress adapted cells. So this would be, I think, a pretty comprehensive approach to how one would characterize a formulation to then provide guidance to the end user. So the key things here is, it's in your best interest to find out some of the details of that validation study, because what one often sees in a firm's record is there's a checklist item. Are you using a validated process? Yes. You go back to the manufacturer. Do you guys have a validation process for what you're delivering to me? Yes. And it stops there. And it is complicated, or can be, and complex. So don't be shy about asking for help. And this is just another example, just to make the point. done, sort of taking the canal water, blending it with fish emulsion or algal extracts. Again, this is benchtop, reactions, but it's been done, multiple times at warmer temperatures, all temperature related. You get a burst of growth. Eventually they drop off and die off, but you can have that short-term growth, and certainly within a tank, and that's been shown, or within sprinkler pipe, or other areas, that those will persist. So this is not coming in with the product in general. This is not coming in with the product, but this is something that you have to anticipate, you have to be aware of, I think, when you're using these formulations. So, one of the other things to really watch for is, like, does the supplier or distributor participate in a certificate program for safety, as well as, verifying the composition, the nutrient quality, the number of microbes that are meant to be beneficial, whatever they're meant to provide, and then increasingly, has the firm been inspected by a public oversight agency? Certainly in California, I imagine Arizona's either doing or moving towards something similar, really looking at many of these products and asking. I know that they're doing surveys now, that probably that data will come out later, but there's reasons that they're concerned. Or is there a site inspection and testing record by a recognized third party, which CDFA has said that they will accept. So again, all of that should provide the confidence in what you're using. I pulled this example out kind of for fun, for a couple of reasons, but it has been used increasingly used, basically domestically and internationally. And you're not actually allowed to label things as fossilized bat guano anymore, but either as pellets or as teas. And just wanted to make the point, there have been a number of studies that, EHEC, or these Shiga-toxigenic E. coli, have been detected in bat guano. Salmonella has been detected in dried bat guano. They actually survive, really well once they're dried down. And the country of origin of where this is being excavated from, bat caves or mined, can be risk relevant, because there is increasing evidence that certain areas have greater potential unless there is a validated process, thermal treatment or otherwise, not just drying. And even because of the different blends and things that have happened, they're starting to look at certificates of analysis for actual bat DNA, as well as other possible sources of trying to adjust the fertility management component of that. So, there's just such a diversity of organisms. So as Shauna mentioned, there doesn't necessarily have to be a formal validation study. There can be credible, scientifically valid study for what can happen. This is somewhat older data from an older publication, but just to make the point, to understand that if a nutrient is added to do what it's meant to do, sort of activate the beneficial, microbial community that's out there, as you add things like molasses, like whey, other products to stimulate that growth, if that product becomes contaminated, or even if it's composted material that has gone through a validated, process to make the compost to then make the tea, we understand from testing, from surveys, that depending on how that's done, how well, and how, the fidelity with which it matches the process that's been validated, there can be residual populations, and you get these bursts of regrowth. The same thing for 0157. So some of the questions is, what challenge study was conducted on finished product to determine the growth potential for foodborne pathogen? What are the specific lot-to-lot SOP verification tests that were done? And again, I see this at the manufacturer end more often than not, sometimes at the distributor, but that's less likely. And then if that nutrient is added for beneficial microbes, has a validation study been conducted to assess the claims of competitive exclusion over relevant temperature and pH ranges? So again, these are just some of the components. Another one of these things that often comes up is that to ask is, if you're going to hold up a validation study to support the product or support your use of it, does the study conditions even match practical commercial conditions. And I see this, periodically, not extensive, but, they say, well, algal extracts, seaweed extracts, other things have been shown to have these antibacterial, antimicrobial activity towards foodborne pathogenic bacteria, such as Salmonella, El Mono, this sort of heat map on the right. The details aren't important, but you can see some were red, a lot of lethality, green, not so much. But, does the marketed product even match the study? In this case, they refer to a study where they made an extract, of these materials and then done a validation test or a challenge study test on the lab bench in a petri dish with a putting the extracted materials directly on a disc for diffusion assay. So how does that even relate to, using it, in the field, in soil, on the crop, whatever it may be? So it's important to ask these kinds of questions. Now, this is about, I think, next to last. So, does the product label information even make sense? I pulled this off of a product, and they listed their microbial analysis of the product, and I don't see anything in there related to assessing or measuring the microbial content of that. So ending now and getting ready to then, tag team hand it off to Cliff. There are various schemes that have been proposed. This is one that's, more specific for Europe. Again, the details here on this graphic aren't important, but you can see, green, ones that have been evaluated for this proposed environmental or human safety index for these materials. Red, there may be some concerns. So should there be, a protocol, a certification, a review, something similar to this in the US? And with that, I will stop and I will turn it over to Cliff to walk you through his comments. Thanks a lot.
Thank you, Trevor. I appreciate it and appreciate the opportunity to be here. I'm assuming, Greg, can you hear me?
Yeah, you're good, Cliff.
Okay, great. Well, if we haven't succeeded in demonstrating that this is more of a witchcraft than it is a science, we haven't done our job today. So with that said, I thought it would be of interest to just take a walk backward into how we got to where we are with validations, verifications. And it kind of started in 2004. And it actually involved 13 million pounds of raw almonds that sickened 25 people with salmonella. And it involved big brand names like Kirkland, Trader Joe's, and Sunkist. And FDA was extremely concerned about raw almonds, as they were with raw commodities, not knowing that, raw commodities at that point was going to eventually turn into produce and the things we're discussing today. But the almond board, because it was such a high-priced commodity and very valuable to California's economy, reacted very quickly and formed what they called the TERP, which was a technical expert review panel. And that technical expert review panel set up processes and validations and identified what they considered to be recognized process authorities. And those process authorities were tasked with going out and validating that, first of all, verifying that the processes that were being done were valid, and then validating that what they said they were doing, they were actually doing. In 2007, the whole concept changed because the FDA had totally outlawed raw almonds for sale in the United States. So everything had to be processed, either by heat treatment or chemical sterilization. So now all of the process authorities had to actually conduct the validation tests on the heat treatments, be it roasting, oil roasting, or dry roasting, and on the chemical treatments, which were mainly polypropylene oxide treatments. What stood out to everybody was the fact that the almond industry had no concept of process controls, HACCP, which is the hazard analysis critical control points, or good manufacturing practices. They were actually just cooking the color if they were doing any kind of process. So the FDA, when they outlawed the raw almonds, said, okay, you're going to have to pasteurize all the almonds in the United States, and we expect no less than a 5-log reduction in the kill of the pathogens. Now that 5-log or greater reduction kind of became the gold standard that most everybody adhered to. And it went as process authorities. So now you turn the clock forward to about 2006, or backward 1 year to 2006, Earthbound Farm spinach outbreak comes about. And having been involved in that, I can tell you that the FDA found no E. coli in the Earthbound Farm process facility, but they did find the E. coli 0157 in the growing fields associated with the crop that was harvested. So the conclusion that FDA came to is what they call RLTO. Was it reasonably likely to occur that the crop was contaminated with the E. coli 0157 in the field? And that was a very reasonable assumption by everybody's part. And as a result of that, FDA kind of turned some interest onto the inputs that would be in soil amendments and the inputs. 2007, Earthbound Farms formed a technical committee of outsiders to review and recommend guide future processes. And part of that was to develop a strategy that looked at the soil amendments, the soil inputs, and they should be validated to a 5-log reduction where possible. So under the validation process, we took a look at how do we challenge outside of a laboratory, how do we challenge processes like pelletizing, like at the time composting, like at the time fish emulsion. Having owned a couple of laboratories, I had the benefit of saying, okay, we can do fish emulsion in a laboratory and demonstrate the 5-log reduction. That's not a problem. But we needed to find surrogates that would be able to be used in a process like a pelletizing operation and use that validated or certified surrogate as a marker for a 5-log reduction of salmonella and other critical pathogens that were associated with it. What I came to the same conclusion that we came to in the almond industry was that once we got out there and looked at how fertilizers were being manufactured and compost was being done and how a lot of the pelletized operations were being done, was that these guys didn't even have a clue that they were in the food industry or serving the food industry. They were just doing whatever they wanted to do. It was actually kind of the Wild West. And for those who are old enough to remember, in 2009, the FBI raided Port Organics because he was just adding ammonia, anhydrous ammonia to the fish emulsion to boost up the nitrogen value. So we've been down a road where a lot of confusion. We had no guidance from the FDA at the time because they really didn't understand the produce industry, let alone understand the fertilizer industry. So at that point, we took a look at all the inputs, and lo and behold, it dawned on us that starts are also a soil input. So having visited a number of the facilities that were selling the starts, the plants that were being planted in the fields, they had no concept of what good manufacturing practices were or what HACCP included. And today, the discussions today all point to the same thing. We tried desperately to validate through technical research papers or through studies that were conducted. And yeah, while we came to the conclusion that we could validate a process of fish emulsion by growing an acid acclimated salmonella and inoculating it into the fish emulsion that was being presented to the growers, it really had no relationship to the way it was being used, and it still doesn't. So the validation was more of an effort to ensure that the people that were making the fertilizers and the soil inputs were the ones that at least could control their end of the business, and if nothing else, hopefully prevent a salmonella or an E. coli from making its way onto the farm through their commodities. You know, what Trevor pointed out, and we pointed out previously, was that products that are validated don't necessarily consider end user abuse or end user use. So therein lies the problem. How do we actually go about trying to anticipate how the product is going to be diluted, how it's going to be used in the farm, how it's going to get abused or recontaminated at that level? And it's almost impossible to address any of those scenarios. But I do have a couple questions that might help us get through some of the validations. I mean, if you're, as a grower, receiving a commodity that says it's being pH adjusted to a level, I would really want to know how frequently you as the supplier, do you even have a HACCP program? Are the critical control points, such as the pH meter, being controlled? You know, do you have an environmental monitoring program around that, so that I know that after you've adjusted it for the pH to keep the control of the microorganisms and the pathogens in check, are you recontaminating it at the other end? I can tell you that when I first went to these operations, the same hose that was used to unload the raw fish was the same hose that they used to fill up the tankers of the processed fish emulsion. So all of your validation kind of goes out the window when you realize that I'm recontaminating everything. And I think the biggest fundamental thing that should be asked of your suppliers is, has a qualified individual done an on-site evaluation of not only your process, but of your risk assessment, of your HACCP program, of your GMPs, and do you have a statistically valid sampling program that you're presenting to me on a COA? If I'm making 50,000 gallons of fish emulsion or 50,000 pounds of pelletized manure, and I come to you and I say it's negative in a 25 gram or 50 gram or 100 gram sample, that's not statistically valid, and it never will be. And it's incumbent upon us as end users to ask these questions and find out really what are you selling me? It's my name on the finished product. It will be my rear end that the FDA comes after. And if you're not asking the questions, you're not doing the due diligence that I think that the subject dictates. Yeah, you know, we didn't anticipate a whole lot of the ways that things were being used or abused at the farm level when we first set out to validate processes. We didn't really have a great array of surrogates to use to validate that if we killed salmonella, was it equally as effective on E. coli 0157H7. And yeah, you can use D and Z values, but one of the things that we do need to understand is that D and Z values actually change as the product dries, as Trevor pointed out with the guano, or as it's subjected to changes in pH. So organisms are unique in the fact that they can adjust and accommodate environmental changes rather well. So what the book says isn't always what the organism has read, and it kind of behaves all on its own. Yeah, then we got, we're caught now with the advent of non-traditional inputs, such as the soy and the blood meal and the bone meal and the things that, how do you actually understand how to inoculate blood meal when it goes through such a high heat process, but yet it can get recontaminated in the end use? How about the bone meal? I mean, you're incinerating the bones at temperatures that we know the organisms don't live, but yet we can find pathogens on occasion in bone meal. And it's all about the recontamination, either at the production level or at the farm level. And therein lies the problem. Which came first, the chicken or the egg on that one? I'm going to turn a lot of this over to Mike to give you a little better handle on what's going on today. But keep in mind that if your supplier isn't identifying his critical control points in the process, and he doesn't have the fundamentals in place to measure those critical control points and to verify that he's actually monitoring those critical control points, all is lost. If you see that they're telling you that the process was heated to a certain temperature, you have every right to ask for the temperature recording charts. And if all they're doing is sticking a thermometer in every 15 minutes, what happens 5 minutes after they took the temperature? Am I still in control or have I lost control? These are all valid points that, as an end user, you want to be aware of. You want to be aware of the fact that there are people out there that can help you perform these validations, be it Trevor or be it the LGMA in general, to assist in answering the questions of what is a good validation, who should be assessing that validation. And with that, I'm going to turn it over to Mike, and I appreciate everybody in attendance and the opportunity to at least go over a few things that we did in the early days, try and make it a more palatable system. Thank you.
Awesome. Can everybody hear me okay? I want to make sure that you guys can hear me okay.
You're good.
You're good, Mike.
Great. Cliff, thanks so much for all of that. And I'd like to say thank you to everybody for allowing me to be able to do all of this. And you really made my job easy. And I can say that from the standpoint that, man, you guys did all the heavy lifting. You were singing my song, and now all I have to do is take that song and bring it into my house. And so that's what I'm going to do here today, give you more of a practical application of what everything that's been said into how we do things at our manufacturing facility. So it's an application of food safety principles on stuff that you already know, HACCP and all of these same things that were mentioned previously. So, and I'll start out by saying a couple of things here. As a manufacturer, we do, as Cliff said, we have to consider ourselves part of the food chain. Our products contact the edible portion of the plant. And that was very key for us to be able to do some of the things that we were convinced some of the people that we were needing to do to be able to get to where we're at today. Understand also that each manufacturing process is unique. The way we do it doesn't necessarily mean that's the way you should do it. It is equipment dependent, it is process dependent, it is the company dependent. And I also like to clarify that this is not a sterile operation, but it is pathogen free. And we're going to do everything that we can to ensure that we can demonstrate that it is pathogen free. Certainly have to have a risk assessment and those mitigation steps. Those are all very simple things to being able to apply a food safety and quality system, AKA HACCP. If it's HACCP or HARP-C or some other food safety quality system that you have, that's great. We use HACCP at our facility, and there's 7 principles there. I'm not going to go over those necessarily in detail, but I do want to point out each one of these things. In the interest of time, I'm just going to breeze right through some of these slides and then concentrate a little bit more on the validation part of it. But you do have to understand your suppliers, your raw material handling, and handling all the way throughout. How good is your equipment and even your factory design? what manufacturing processes you use, and how do you document that. It's all part of the HACCP system to show what you do and what you're doing, what you're saying that you're doing. So I'll just briefly go over the supplier side of things, how we do it here at our facility. We evaluate our suppliers, and just as our customers come to evaluate us. So there is some level of supplier approval that needs to happen. On a number of occasions, we've had our customers come and visit the plant, which we're more than happy to do. And we want to be able to demonstrate to them that we are actually doing this pathogen elimination. Now, we'll note that some of our suppliers that we use, we are expecting them to provide a quality product. That doesn't necessarily mean that it's pathogen free, but if there is a heat treatment process like the blood meal and the bone meal that Cliff had referred to, there is a heat treatment process. So it should come with a C of A, and it should come with an understanding that they're handling those finished products to them, their finished products, in a manner that will prevent that cross-contamination. For us, that's not good enough. So everything that we're bringing in, which includes the blood meals and the blood meals, the compost, the protein meals that have been rendered, all of those things, we're considering those to be contaminated. So not until it goes through our heat treatment process do we consider that a clean process. So we handle those things separately. So all of the raw material is handled in a different area. It's got different equipment. It's got a different storage. It's got different people that handle those same things. It's part of the factory design. How does your factory or your facility minimize cross-contamination? Do you have separate processes for the raw material like we just mentioned? Is the processing part of it? How do you separate that? And is your finished product, how is that treated? And certainly for us, that is one of our most guarded places, is the finished product, where we do have separation of traffic and separation of equipment. It is a separate building. We have separate loaders and also separate people for that. So let's get into the nuts and bolts of it. And I know we're limited on time, but I do want to talk a little bit about this validation. It's very simple for us. We're looking at these target organisms that are outlined as part of 7C in the updated metrics, and have been in the metrics for a long time. Fecal coliform, salmonella, S-Tech, which includes E. coli 157, Listeria monocytogenes. For validation for our pellet process and also for a liquid process, the question is, can I kill these organisms? Not everything else, not going to be sterile, but can I kill these target organisms? Our goal is to be able to quantitatively show that yes, we can absolutely do that. Is it a 4 or 5 log reduction? It's at minimum 4 log reduction. And is there a standard out there right now that says what is the recommendation for that reduction, that log standard? We can also consider a surrogate. We're actually using those same materials for that. And it really boils down to time and temperature. How long does it take to kill those organisms? And what is the dwell time inside that operation? So what I have pictured onto the left is a pellet mill, and it's one that we use. And one of the things that Cliff had talked about earlier is, how do you capture the heat that's generated in the pelletizing process itself? Well, for us, we don't even consider that. It's hard to measure that. It's hard to even take a look at that. But what we can measure is the conditioner where the steam is introduced. Can we monitor that? Do we have a thermocouple that we can use in real time to monitor that temperature? Can we call that our critical control point? Absolutely, yes. That's something that we do. Is everything else behind that a bonus, like the heat that is generated at the pellet mill, where there is some frictional heat and some pressure that is generated? Does that kill? Absolutely, but it's not measurable for us. The way we validated our process was to be able to take our raw material and then count it at the beginning, and then go through the process, catch it after the conditioner, and count it afterwards. To me, that is the most solid validation that you can get. you're counting on the material that you're using, going through the equipment that you're using, and then count it at the back end. Ultimately, if we change the time and temperatures and look at that, we could, I guess it's more like, without formally doing it, we are generating some D&Z values. We were able to demonstrate that reduction. And even further, we were able to monitor it on different pathogens as well. So for us, all the different ones that our greatest risk, we think, is Salmonella. We're able to find some raw materials that have a high count for Salmonella and demonstrate a reduction there. But then the question becomes, well, what about fecal coliforms, or what about LM or any of the S-Techs? Can your equipment, is that robust enough to be able to kill that? And can it kill it on poultry manure, or can it kill it on a protein meal, and any combination after that? So it starts to get really complicated after a while. And at the same time, we want to make sure that we're have the ability to answer those same questions, because this is just part of the story. In the grander scheme of things, the worst case scenario was that there was a recall. It was traced back to one of our products. I want to be able to have a story that says, yes, we can, our equipment can kill this material, and here's how we prove it. One way to do that is this third party that Cliff's referring to, is a process authority and or a laboratory that can do that. So that's exactly what we did. We brought a laboratory in and said, hey, here's what we're wanting to do. Count it up front, count it out back, and we'll show you that. Let's see where the data lies out. Turns out we were successful at that, but are we successful at that every single time? Which is where the verification comes in now. Verification is that exactly what Chana had mentioned, and I'll word it in a little different way. Can you achieve the time and temperature, and can you document it? So more specifically, are you doing the right thing, and how can you prove it? So we do that every single day and in every single lot. And I'll show you this thermal profile where we have this operational limit and this critical limit. So that red line that we've established there is that critical limit for our manufacturing processes. If there is a time that any of these materials or these pellet mills will go down below that, then all the lights go off. Everybody gets notified. At that point, for every production, we have to prove that it wasn't contaminated. There are occasions where during the process, you'll have to maintain that pellet mill and shut it down. Can you document and show that it wasn't producing any material? Otherwise, there's some real answering that you have to do. It's certainly a corrective action and a non-compliance for us. But in normal processing, everything happens above that or around the operational limit, never to go below that critical limit. I'll also offer up another set where if you look backwards, you can take a look at what you've done and say, how long have I been able to do this at this amount? And am I able to demonstrate historically with the number of CFAs that I've done to show that the process is working? And certainly you can do that. I'll touch briefly, I know we're running out of time. I'll touch briefly on sampling because I think it is important. To the example that was given previously, if you're making 50,000 pounds out of it per run on a pellet mill, is 12 points enough? Absolutely not. For us, we are taking with an auto sampler off the line every 2 minutes. That is a representative sample for us. It falls within the new guidelines for updated LGMA metrics for N equals 60. We do have certificates of training for everybody there. But more importantly, all this heat treatment that you're doing and the sampling, representative sampling that you're doing, it has to be traced back to a lot definition. So in the event that there is a recall for us, we can say, here's this lot number, and here is the heat treatment that we went back to it. And here's the heat treatment, and here's how many times we sampled it, and here's who sampled it. It's a complete story for everything. We are in a hold and release basis. We want to make sure that we are a contact surface. All the contact surfaces are addressed, which means We store it in the right place. We clean and sanitize prior to. Are there challenges for that? Absolutely. We use a bunker and we can easily document the sanitation of that. Starts to get a little bit more difficult in the vertical, not saying it's impossible, but certainly more challenging in the vertical type storage in silos, but it should still be on the hold and release basis. In each bunker, we do not mix lots, and you've got to follow it up with documentation all the way through the entire process. In the end, if there is a recall, we have a packet that shows from start to finish, including heat treatment, including the inputs, including the training. All of it is available for review for that. And you got to be able to count on your people to be able to accomplish that. With the understanding that there are no process authorities right now, we always are looking at being able to prove that to somebody and not the FDA. We want to have somebody that can say, hey, we're going to come into your facility and evaluate you on your food safety and quality system. Without the availability of something like SQF or GFSI for fertilizers, for biological soil amendment type fertilizers, we had to look out for other people, and we found ISO 22000. From their website, they say whatever the size or product, all food producers have the responsibility to manage the safety of their products and the well-being of their consumers. For us, that made perfect sense for us. Our products may come in contact with the food being produced. Thus, we share that responsibility to manage our food, manage the food safety of our products and our well-being of our customers. That's why we chose ISO 22000, at which until the time there is a process authority that we can go to be able to say, here's your certificate, you're good to go. This is what we're going to go with to demonstrate to everybody that we have a documented process for that. And I'm open to any questions.
Thank you, Mike. And thanks very much to all of our speakers today. That was just very informative, and we appreciate you sharing your knowledge and expertise with us today. I'm not seeing any questions in the Q&A, but I have a few for you guys, if you don't mind. Greg, I want to start with you. Would you give everybody in attendance a quick review of what documentation should they be receiving, or what documentation is required in the new crop input section, mainly since we've kind of excluded soil amendments from the presentation today?
Yeah, sure, Vicki, and that's a great question. So as far as the overarching, the LGMAs currently do not have like a template or some sort of a document that stipulates exactly what we need. But when you think about, I'm going to pull up something here that will help. Just give me a second. That will help with the kind of general understanding of what is expected. We have, give me a second here, sorry. Things that we would like from an auditor's perspective, and it kind of goes to what Cliff was mentioning, it really goes to what Mike was mentioning. The documentation that should be received goes into a few high-level bulleted items, right? You want to have something that basically tells you as the grower or the handler what is the product that you're purchasing. The company should be able to provide you with a general statement of what this product is, so that it's clear that when you say I'm having chicken pellets or something of that nature, or bone meal or a tea, what is that product? You should be getting some sort of information from your supplier regarding contacts. Who are these folks that are providing this material? Who do you contact if there's a question, or if you have a challenge, or you have an issue? A little bit of like general information of the product again, just what is this product? What kind of parameters are used to produce this product? So as Mike mentioned, right, it's really important. Again, we're not talking about a Library of Congress type dissertation here. What we're basically saying is a very quick summary of what does this product go through to make it at its finished step, right? So you have a name of this product, you have a general idea of what it is. Give me some idea of the parameters that it goes through, the process that it goes through. Okay, some information about the equipment, you know, and then really the underlying part, again, what Cliff mentioned, what Trevor mentioned, Shanna mentioned, right, is what is the study that was conducted, and then how are you verifying on an ongoing basis that this is being achieved, right? So you get a lot of information, as Mike mentioned, and we updated our metrics that every certificate of analysis that comes with a lot product, the lot needs to be described, and that's really important because of the traceability aspects of the product. If there was a problem, we want to be able to go back to that manufacturer and get more detailed information. But what we want from them is a summary of their process, what they did, you know, the methodology. Did they use certain microbial strains for the study, or did they use a research study that, as Shanna mentioned earlier, right? There's many different ways that you could potentially get a documented validation. Now, Mike's examples, you know, really do amount to a gold standard. Some of the things that might be available from his company really are, you know, the high level of what we like. And then, of course, the idea is you'd want something, like I said, a COA, a certificate of analysis, that just stipulates this lot went through some sort of a verification test that shows that the process was in control when it was produced. And to Cliff's earlier point, we don't require this necessarily. We require pathogen certificates of analysis, but it may behoove the user to also get some other types of analysis, you know, qualitatively speaking. So you know that the products are meeting a certain, again, a pH or a certain other result that really will show that the process was being followed and is verified.
Thank you, Greg. There is a question for Trevor. Trevor, can you explain background activity in analytical results and if their role in log reduction is significant?
So let me take a swing at the question, and if I'm not addressing what the individual is asking about in terms of background activity, you can restate it, and either I or somebody else on the panel can help with that answer. So my initial reaction to background, you know, activity, you know, perhaps, you know, the focus relates to you know, other things that may also be there that will influence the ability to accurately and sensitively detect residual pathogen, you know, survival. So again, all of these things go into the strategy for how you would recover, whether, you know, there are recognized resuscitation steps, and, you know, and sometimes, you know, how long you would allow something to go through that and then go through the growth to meet the particular test method that you're using. Often one sees, you know, validation studies where there is a growth, a process, you know, treatment, I'll just say heat, because it's easy, and then there's a recovery, and, you know, 24 hours later, you know, the you know, reported result is, you know, non-detect or zero. But we know that if you had left that for 36, 48 hours and then used a molecular, you know, approach, you would be able to demonstrate that there is still viable cells present, or other types of background activity in terms of the constituents. I'll give kind of perhaps a funny off-target or tangential example for this, because it came up just, you know, the other week, where, you know, it's not a soil amendment, but, you know, people are interested in using these lytic phage, these, you know, viruses that attack things like E. coli 157. And, you know, the validation study demonstrated, you know, 5 log reduction, you know, after treatment, blah, blah, blah. But the, you know, the real question was, you know, is it really the process of recovery that's causing the lethality, as opposed to, you know, being lethal to, you know, a leafy green, you know, post-treatment? So it doesn't actually happen until you go through the lab process of washing it off, and now you've mixed the phage and the bacteria together in a way that they would die. So I have no idea if that's even close to what they meant by background activity. Maybe Cliff or Chana, you know, Mike, you know, do you have a different spin on that kind of answer?
I agree with you.
Oh, that's dangerous.
Just-
Okay, that sounds great.
But
I have a
again,
question.
I'm not sure if that was the background activity you were talking about,
Oh,
or
Jen,
something else
okay.
completely.
Yeah,
All right,
I know that-
Jen.
I'll just add one more comment,
Yeah, great.
and we put a lot of pressure on third-party laboratories or other research scientists, for example, to really be able to describe the methodology that they're using and understanding how well they work for recovering organisms and what their detection limits are. And I think that's a super important concept to think about related to this, because if you're looking at a very, very small volume, for example, and not very frequently, and you get all non-detects, that means something different than looking at a larger volume and more frequency to be able to decrease your detection limit. So anyway, you should be able to put pressure on anyone who's got a validation, procedure to ask them about those methods, and do they really understand what they're looking for and what they're getting out.
Okay, I would like to ask each of you or any of you that would like to respond. One of the new metrics that we added this year was regarding storage. And once some of these products have been stored greater than 1 year, there's an expectation that the product be retested for the acceptance criteria, which are those pathogens of concern to us. How, in terms of today's presentations, how do you see storage as being a challenge to recontamination, or whether or not those are actually challenge studies that we should look for with regards to long-term storage of some of these products?
I think there's an expectation on the part of the manufacturer that the product is going to be stored at least in a condition that doesn't jeopardize what was delivered to them. So, for example, if you've got drums of fish emulsion and you're leaving them open for any length of time, that validation study is totally irrelevant at that point. The same with fertilizer, if pelletizers. If you're getting a bulk load of pellets and you're using a front end loader to move it around that you just used for raw manure, you can't hold the pelletizer company liable for that. I mean, there's got to be a lot of injection of common sense here and accountability. But yeah, in general, if it was negative when we delivered it to you, one would assume that it's going to be negative throughout the shelf life of that product.
Yeah, let me just jump in, and then Mike, because I'll refer back to a number of the elements that Mike shared within his presentation. I think hold true, and agree with Cliff, but in my mind at least, it's a little bit like the difference between verifying on the microbial side of things, or monitoring that you've satisfied the process controls. It's to me, it seems more satisfactory, to be able to document and verify that you're holding these products, under condition that wouldn't expose them to contamination. I know that's not always possible, but so again, we've all certainly seen it, where bulk pellets are delivered and they're sort of field side stored and in open forum. I think that's whether you test it, again, you can't, how do you sample enough to know, et cetera, et cetera. So I think it's really exercising that control, particularly for things that would be delivered in sealed containers, not open close, something like that. John, Mike, go ahead.
Sure.
Yeah, absolutely. So again, just reinforcing everything that you all have said, is our expectation would be to maintain that food safety integrity throughout that, let's call it the chain of custody. So as supplier, we would certainly provide the information demonstrating that it is free of pathogens based off of our documentation. But then how is it handled? And I'll refer back to something that Trevor, that you had mentioned. Use the term end user risk validation study. Does that apply here? And to me, it's mostly, I'll give you an example of one demonstration I think that we've done before was in our mock recalls. We do a mock recall regularly. We've extended that out to some of our customers where we say, okay, so here we're doing a mock recall. Can we do one in conjunction with you guys as a customer and see where things shake out? This was a challenge, I think, from the LGMA earlier this year, where we have to start sharing some of this information. Again, Trevor, you pointed out the role of the manufacturer and the distributor, the applicator, the grower, all the way through that whole process. That's part of the supply chain. So for me, the expectation would be certainly to store it in a manner that was clean and dry and preventing the cross-contamination, everything that we expect, that our customers expect of us as it stands now within our own facility.
Okay, guys, we're nearing the end of our time.
Do you mind if I just jump in with one
sure,
thing related to that? So
Yeah.
at the either the risk or the likelihood of further making myself unpopular in some quarters, I'm not, just listening to that as a new provision, in terms of retesting product, especially if you've satisfied the storage conditions that are reasonable to prevent recontamination or cross-contamination. I do think it's worth considering taking steps and measures to really verify the application equipment as the place where you would want to know, between lots and over time, whether something has happened within that, because we know many of these products can support the growth of human pathogens, particularly Salmonella, because of their very diverse genetic ability to use a lot of different nutrient sources, particularly carbon sources. That isn't a contributing factor to some of what we've been seeing over the last several years. So maybe not so much the product, but how it's being used, how it's being applied, given those risk assessment validation studies that show this is possible under these variable conditions. So that was just my little.
That's a great point.
Great. Okay, I have one last question for everybody, and that is, if you want to answer it, what would be your top take-home message for our attendees today? And I'll start with Chana, and we'll just kind of go around the horn.
Oh, you caught me off guard.
Sorry.
It's okay. I'll go first. Yeah, I think as you heard all of us kind of discuss, there's a lot of variables that go into play. And don't be afraid to ask questions and put pressure on the distributors or the companies that you're buying products from. It's their job to be able to produce, documentation for you to use. And if they don't have it or aren't willing to do it, maybe it's time to find a different supplier and a different product to use.
Cliff.
I can only echo what was just said. You have an obligation to ask the questions. You have an obligation to answer those questions as the supplier of it. When I would validate operations, we did it with surrogates or we did it in the laboratory. We always explained what the methodology was. We tried to make sure that we got 3 successive negatives before we said, yeah, everything's fine. And while we never did do a whole lot with the consumer abuse, we knew that abuse could happen. So I think what we need to do is educate through questions, educate through webinars like this, how to properly handle some of these things. What is a process authority supposed to do to help make your life easier as an end user?
Great. Trevor?
Yeah, sure. I think my biggest message, takeaway is that, I always say it the same way. I mean, it is in, the producer's best interest to understand, what inputs they're bringing to the farm, to their operations, how, what the potential is for those materials in this category to support growth of environmental contamination, which you're not going to fully, control or prevent, and how that changes under specific conditions, so I think, again, besides, asking what the manufacturer distributor is doing, just how could that, potentially impact, you getting involved in either a recall, which is much more likely than an outbreak, but either one.
Great. Mike?
Good point.
Same for me, really. It's just same as everybody else. Just get a better understanding of the entire process. Don't be afraid to ask the questions. We're here to answer those.
Great. Greg, we'll end with where we started with you. Your last
I just
thoughts.
want to, before I get hung from suppliers who are enclosing their materials, the retesting is only for exposed products, not for everything. So,
Oh,
but
right.
anyways,
Sorry.
yeah, no, that's fine. I just, let's clear that up first before,
Yes,
everyone
thank you.
comes
Sorry about that.
to
Yeah.
town. First and foremost, I just want to thank the panelists for this awesome dialogue and for bringing your wealth of knowledge for us and to the industry. I mean, I learned a lot today as well. I'll be honest with you. I thought I knew, I've done a lot of validation audits myself in a previous life, but that's why, again, I mentioned it earlier, I call Cliff and Trevor and Mike and Channa and, all the time when I have questions about these types of things, even though I've been doing it for a long time, because they're a wealth of knowledge and we're always learning. And I think that's the take home message from me for our industry is don't be afraid. We don't know everything. You can't know everything. This is complicated stuff. And really, from the LGMA perspective, we're going to continue to try to improve your knowledge base as much as we can. But you really do have to, to Cliff's earlier point, take some accountability and really start marching out there yourselves and start learning, get your feet dirty and look at what's going on. I think that's just the best way to learn how things are being produced and the potential impacts on your operation. So that's just my real take home message. But super grateful for the panelists. I don't know, for the audience, we gave them a very short time to put it all together. So you did a great job, all of you. So I really appreciate it.
Yes, we really appreciate it. And we appreciate everybody that attended today. And we're very grateful for our speakers. And I don't see any more questions in the Q&A. And so enjoy the rest of your day. And obviously you can reach out to Greg and I with your questions regarding specific metrics, and we'll try to get your questions answered or get you directed to the right person or place to help you with that. So thanks again.
One last thing, I just want to wish everyone a happy holidays.
Oh, good idea. Thank you. All right. Thanks, everybody.
All right. Have a great day, everyone.
Bye-bye.
Bye-bye.
Thank you.